Scientists found mechanism that may affect COVID variant infectivity. COVID-19 variants remain a mystery to scientists. Although they know that mutations let the variants infect more people, the specifics remain unclear. Recently, scientists found a mechanism that may affect the infectivity of these variants. But before we continue, please like this video if you find this informative and subscribe to our channel for more interesting and informative content. Don't forget to click the bell icon too to get the latest updates. Kelly Tenhagen, senior investigator at NIH National Institute of Dental and Craniofacial Research, led the new study published in the journal PNAS. Researchers first detailed what variants are. They explained that variants are similar to the original strain except they acquired mutations. With these mutations, the coronavirus gained better chances to spread and infect more people. The first variant that made an impact was the Alpha. It was later replaced by Delta, which is now the most dominant in the world. The latter was the reason why vaccine boosters were rolled out in many nations. At a closer look, the coronavirus has spike proteins on its surface. These are needed to attach and infect cells. Yet the spike proteins won't do anything until they are activated by cleavages of host proteins. The furin enzyme is among these host proteins. Variants have several mutations on their spike proteins, which can influence the cleavages. Researchers said that mutations in Alpha and Delta variants seem to improve the furin cleavage. However, there have been cases where excessive sugar molecules could lower the protein cleavage. The GAL-NT's enzymes could ramp up these molecules. The team studied the GAL-NT activity on spike protein using fruit fly in mammalian cells. GAL-NT1 was found to add sugars to the wild spike protein, and that lowered the furin cleavage. But mutations in Alpha and Delta overcame that activity and reversed the effects of GAL-NT1. This suggested that GAL-NT1 could partially suppress furin cleavage. In people, they analyzed the RNA expression in cells from healthy volunteers. A wide expression of GAL-NT1 was found in lower and upper respiratory tract cells, which are vulnerable to the coronavirus. So GAL-NT1 plays a role in SARS-CoV-2 infectivity, and if it can be tapped, scientists may unlock new treatment or vaccine designs against COVID-19. However, the research is still in early stages. Do you agree that some people are more vulnerable to COVID-19 than others? What do you think they should do to lower their risk? Share your thoughts in the comment section. Till our next video, thanks for watching.